Hello. My name is Amy Bastian, and I'm going to be giving the next 2 lectures in your course. We're going to be talking, first of all, if you're in the back and you can't hear me well, let me know, because I don't have a microphone. I have a lot of microphones, but none of them actually project. So if you can't hear me, let me know. I'm going to teach you 2 lectures on subcortical structures in the motor system, and today we're going to cover one of those structures, the cerebellum. So what I'm going to do is walk you through a series of experiments and talk to you a little bit about anatomy and show you some information from patients. I should just tell you briefly, I work on cerebellar patients. I work on lots of different kinds of neurological patients. My laboratory is at the Kennedy Krieger Institute at the med school campus, and my interests are really in understanding movement control and learning well enough to be able to help with rehabilitation of these kinds of individuals. So today we're going to talk about the cerebellum and the basal ganglia. They're both subcortical motor structures, and the first thing I want to emphasize to you is that these structures impact movement control through their projections to other motor structures. There aren't direct projections from the cerebellum down to the muscles via the alpha motor neurons. There aren't direct projections from the basal ganglia to the alpha motor neurons. Instead, they influence movement through their connections to other motor structures. So, who can tell me what motor structure this one is? This is a no-brainer. No pun intended. Motor cortex. Very good. Okay, so primary motor cortex is one of the target structures for the cerebellum and the basal ganglia. They both project through the thalamus to get to the motor cortex. And of course, the motor cortex influences movement through its projection down the spinal cord via the corticospinal tract to ultimately influence muscles and make movements. There's another structure in the midbrain called the red nucleus, and this structure also has a projection down the cord called the rubrospinal pathway. It really only influences head, neck, and arm muscles, but the cerebellum and basal, mostly the cerebellum connects to the red nucleus. I will correct myself. Now there are 2 brainstem pathways that these structures interact with as well, the vestibulospinal and the reticulospinal systems. Oh, let me back up. By the way, I have created a sort of a mini chapter with lots of text and figures. So I will go a little fast sometimes, and stop me if you have questions, but know that in that document you'll have a lot of this information, okay? Okay, so these pathways are from the brainstem. They project bilaterally, and they're important for controlling things like upright posture and gait. And so again, this is another pathway by which the cerebellum and the basal ganglia can influence movement. Now, I want to talk today about the cerebellum, and I want you to consider the control problem that we're dealing with here. The motor system has to be able to move the plant effectively, and by plant I mean your body. Now, it's not a simple body. It's not the most complex body, but it's not simple. You have many hundred muscles, 600 muscles, about 200 bones. They're all interconnected by lots of joints. And so the dynamics of this system are actually fairly complicated. Each joint isn't a simple joint. Think about your shoulder. Your shoulder is a ball and socket joint. It has many degrees of freedom. You also have muscles that cross more than one joint and that run in interesting directions. And in addition to that, we know that muscles are nonlinear force generators. So the signal you put in is not exactly the signal you get out. Muscles are low-pass filters. They damp things. This makes the system, you can imagine, these are your controllers, these are your effectors. They're rather difficult to control in sort of engineering terms. There are also mechanical interactions with parts of your body. So when I start moving my arm around, it actually affects my trunk, and I have to stabilize against that force. Now, here's one of the big problems with this control system. We have sensors that are pretty good, vision, proprioception, our vestibular sense, but they're noisy, and we have to integrate across them. So we have to be able to make sense of what we're feeling and what we're seeing and match it all up. In addition, we have to be able to use this information even though it's out of date. It's very slow. And so what I like to tell students is that right now, everything you see, everything you feel happened in the past. Nothing you are seeing and feeling is current. It's all happened at least 100 milliseconds ago. So you have no up-to-date information. You have nothing online right now that is happening in the moment, which is kind of cool in a philosophical way. But what that means is that your motor system doesn't have access to your current state, to the current configuration of your body, to what she's doing with her computer over here. You don't know exactly what it is. And that slow system makes it very hard to control movement. And so we know that our movements have to be fast, in some cases very fast, if you're running away from a tiger or a mean parent. Adaptable, we have to be able to change them. They have to learn. We want movements to be automatic. We don't want to have to think about everything we're doing. And we want to be able to learn things over the long haul. So this is why I think movement control is incredibly cool and interesting, and there are a lot of good things to study. Now, the cerebellum, we think, helps to solve some of these problems, and that's what I'm going to try to tell you about today. So to start with, I'm going to show you somebody with cerebellar ataxia, and I'm going to show you two patients, one reaching and one walking. Now, before you look at these guys, I want you to know up front, damage to the cerebellum doesn't cause paralysis, it doesn't cause weakness, but it does cause motor problems. It causes something that we call ataxia, which is movement incoordination. Now, how many of you have ever seen somebody who's drunk? All right, yeah. How many, well, I'm not going to ask the obvious follow-up question. But when people are drunk, they become clumsy, right? They stagger, they veer, they may miss the drink they're trying to get when they reach. And those problems look very much like what people with cerebellar ataxia have. So let's look at our first guy. He's going to be doing a task where he's reaching to into his chin and my finger. And you're going to see that he can get there, but his movements are irregular. So let's look at him first. So you see how he's kind of swerving and missing? He missed his chin, he missed the finger. And you'll also notice his trunk is kind of wiggling around as he's moving. He's not stabilizing it very well. Okay, now let's show you somebody who has an ataxia when they're walking. So this is a guy who exhibits the sort of classic features, a really wide-based gait. The legs are kind of spread apart as he walks. He kind of has to catch himself. We're having him walk around these cones. So he can walk at this point, but he's clumsy. He looks like a drunk person. This is exactly what goes wrong with cerebellar ataxia. And so the engineering question that's so interesting is, why do you get this problem? What's happening? And one of the things that we think has happened is that the cerebellum is normally sitting there making good predictions about movement so that you have a shorter loop time for online control. In other words, the cerebellum is doing its best to make some predictions about how your motor commands may unfold, and in doing that, if you lose it without that prediction, you have to try to rely on time-delayed feedback. So you remember I told you you're living in the past. Well, if you don't have a good predictor in your brain about how your movements are going to unfold, what you have to do is you have to depend on that slow feedback. And we think that's one of the problems that's gone wrong here. So if I slow this gentleman down and show you exactly what the timing might look like, there he touched his chin, he made a correction, and now he's getting feedback that his finger is here when he's actually here. And so what that means is that he keeps moving, and he overshoots the target and has to make corrections. So this is one prominent hypothesis about what the cerebellum is normally contributing to movement. It's doing some kind of prediction. Now, when we think about predictions, I want to just emphasize why this is so important. You need it because of this complex dynamic system that I told you about your body. So if I stand here and I lift up my arms like this guy, sort of, as fast as I can, what muscle do I turn on first? Anybody have a guess? Legs isn't a muscle. Point to a muscle. Pick a muscle. Somebody. Come on, it's more fun for me. Alexandria. You picked that one? Okay. Anybody else? Tongue. Wrong. Next one. Oh, come on. Okay, the obvious one is this, right? Your anterior deltoid. You might say, oh, you got to turn on your back muscles because your back's got to be stable. Well, Alexandria is close to being right. Wrong side of the leg. But this muscle here, this is called your anterior tibialis muscle. It's the muscle in your shin. You turn that on 50 milliseconds before you turn on your anterior deltoid. And you can record this in the lab. I mean, it's dead easy. Throw some EMGs on people, have them make a fast movement. You can see the timing. This is a learned response. This is something that the nervous system learns that you lose when you lose the cerebellum, and actually that you don't have when you're a little baby. So little babies also sadly look drunk. They wobble and they have a wide-based gait. And when little babies pick up their arms in excitement when they're standing up, little toddlers, what happens to them? They fall over. They fall over backwards. They fall over backwards because they don't have this learned prediction that helps them to control their body. So we think that the kinds of things that do this in engineering terms would be called an internal model. And so David Robinson, who was here at Hopkins, who you've probably heard about before, because he really was the person who used engineering principles to understand eye movement control, he said that he thought that there had to be some kind of internal model to steer fast eye movements, the CADs. And so this is from a book by somebody you probably know, Reza Shadmir, who's just quoting what David Robinson said. And so there are two kinds of internal models. And the reason I'm telling you this is I'm going to show you some physiology about this. So there's an internal model where a motor command might go through a forward model to predict kinematics. So in other words, I might be sending a command to my muscles, and this model will tell me where that command will take me, where my arm will go. There's the opposite kind of model in engineering, an inverse model, where you say, I know I want to go from here to my water glass. And so what muscle activities or what forces would I need to generate to get that? So you can have two of these kinds of models. And of course, biologically speaking, you're probably not implementing this exactly this way, but this is one way to solve this problem of having an internal predictor, is by having a model sitting in your brain doing this. And so we're going to talk about some studies that have looked for neural signatures of these things, or that at least can be interpreted in that way. Okay, so now everybody loves movement control, right? It's cool, it's exciting, the cerebellum's great. So now you have to slog through the anatomy a little bit, which is usually less fun. This is the cerebellum. I've peeled away the rest of the brain. Well, I didn't, Netter did. And what you're just seeing is a back view of the cerebellum. And the first thing that you'll notice is it's highly folded. It's much more highly folded than the rest of the brain. It's made up of really three lobes. An anterior lobe, which is this part right here, large posterior lobes, and then there's another little lobe in the front called the flocculonodular lobe. So by lobes, I don't mean right or left. I mean these three anatomical lobes. And the reason that we divide it that way initially is based on phylogeny. So if you go to lower animals, they have conserved these lobes and actually lobules within them. Okay, so that's sort of gross structure of the cerebellum. Now when we think about function of the cerebellum, we actually think more about it in terms of longitudinal zones. You have a midline part of the cerebellum called the vermis, because it looks like a worm is laying on the surface of the cerebellum. Then you have an intermediate part of the cerebellum and a lateral part of the cerebellum. And that's the way I'm going to ask you to think about this, because it makes much more sense in terms of the circuits that are running to other parts of the brain. Now, the cerebellar cortex is this really highly folded structure, and it's packed with cells. It's packed with only 5 kinds of cells, but it's really packed with cells. In fact, the cerebellum has more cells in it than the rest of the nervous system combined. So there are a lot of neurons there, which means it's got to be learning and encoding something that requires a lot of information storage. That's what we think. The other thing that I'll point out on this slide is that deep in the cerebellum, you have the folded cortex, and then deep in the cerebellum are these nuclei. These are called the deep cerebellar nuclei, and they run from medial to lateral. And this is the, the vestigial. Two nuclei called the globose and the emboliform. In animals, we call them the interpositus, and that's what I'll tend to call them here, I guess in non-human animals. And then a large dentate nucleus, which there kind of looks like a weird tonsil or something, but it's a highly, it's a large structure that's sort of highly lobed and folded. The nuclei are the output of the cerebellum, and so we'll talk about that in a minute. Okay, here's a ventral view of the cerebellum. So we've cut it off the rest of the brain. It was attached to the brain through these peduncles, which are sitting here. There's a middle peduncle, an inferior and a superior peduncle, and that's how information goes in and out of the cerebellum. So those are white matter pathways, axons. Here you can see the flocculonodular lobe. It's kind of a weird looking little lobe. It's this piece right here, and then this and this. So it's this sort of squished thing on the ventral surface of the cerebellum. Okay, so that's the general layout of the cerebellum. Now the other thing that I didn't mention, and I'll tell you more about later, is the cerebellum only has 3 layers in the cortex. How many layers does the cerebrum have? 6, yes, 6. This is 3. So that means it's twice as efficient, right? No, kidding. Okay, okay. So I told you about the anatomy, and now I'm going to tell you about how I really want you to think about the anatomy of the cerebellum. I mentioned earlier that we can think about anterior, posterior, and flocculonodular lobes, but what I really want you to take away is that you should be thinking about sort of circuits. And those circuits run medial to lateral, and they involve connections from the cerebellar cortex to the deep nuclei, which then project out to influence other motor structures. So again, no cerebellar projection down the spinal cord. So the midline part of the circuit here gets input from the vestibular system, both primary afferents and more processed information, the reticular nuclei and the brainstem. It gets direct sensory inputs from the body. It's through pathways called the spinocerebellar pathways, and it gets visual input. So the inputs are from the body, vision, the vestibular system, and the brainstem nuclei. This information projects into the cerebellum, and it projects both to the cerebellar cortex and to the nuclei, the most medial nuclei, and then it projects out to the vestibular and reticular nuclei in the brainstem. So based on what we've talked about so far, what do you think that this kind of information might be good for controlling? Based on the anatomy, what kind of movements? Do you use your vestibular nuclei to make reach and grasp movements? No. What do you use those brainstem structures for mostly? Posture and gait, yes. So this circuit has, and if you damage this circuit in a very specific way, we've studied this in humans and in monkeys, you get really bad problems with posture and gait. But you don't have problems with fine hand control or reaching. So we think that this part of the cerebellum is particularly important for that. There's also a lobule in here that's important for making appropriate magnitude saccades, calibrated saccades. Okay, the intermediate cerebellum gets input from only 2 places, the spinocerebellar pathways from the body and a corticopontine input. That's from the cerebral cortex to the pons to the cerebellum. It projects back out via the thalamus to the cerebral cortex and specifically motor areas, motor cortex, premotor cortex, supplementary motor area on the other side of the brain. Now, in this case, we'll just say right is right. If the right cerebellum is projecting to the left motor cortex, what side of the body is the right side of the cerebellum controlling? Say it again? The right, that's right. It's ipsilateral control, right is right. So right projects over to the left cortex, and you remember the left motor cortex crosses when it sends its projection down to the body. And so you have, when you damage the right cerebellum, you have symptoms on the right. Damage the left cerebellum, symptoms on the left. This projection, this circuit through the intermediate cerebellum is important for controlling fingers, hands, and arms. It's important for controlling the kinds of things that you do with motor cortex. So reach to grasp, finger manipulation, voluntary kinds of movements. Now the lateral cerebellum is big, and it only gets input from the cerebrum and lots of cerebral areas. It gets input from parietal cortex, from frontal cortex. We're finding out that it gets some temporal inputs. It gets inputs from all kinds of places, and it projects to prefrontal cortex, to the parietal cortex, and also to some motor areas. But this part of the cerebellum is pretty interesting because we think that this part of the cerebellum is actually influencing non-motor things. So people have studied a variety of different kinds of language tasks, spatial working memory tasks, and you can have subtle deficits in those tasks if you damage this part of the cerebellum. Now, if you see a patient who has a cerebellar lesion, their movement problems will immediately be obvious. And these cognitive things will not. And so it's much more, we think that the control that the cerebellum exerts is much more necessary for the movement control system than it is for the cognitive system. Now, this is the ventral part of the cerebellum. This is the flocculonodular lobe. It gets vestibular inputs from the canals and the otoliths. It gets vestibular nucleus information, and it gets visual information. And it doesn't have a deep cerebellar nuclei. Instead, it sends its projection from cortex directly out to the vestibular nucleus. This part of the cerebellum is important for balance control as well, but it's also important for calibrating a reflex. Does anybody know what this reflex is? If I stare at you and I move my head, I can still stare at you. Why is that? Do you know the name of this reflex? Really? Come on. Vestibulo-ocular reflex? Wow. Yeah, no, that's the answer. I'm sorry. Is that a question? No, that's the answer. I'm sorry. I figured you guys had covered this already. Cool. Okay, let's do a little demo then, because this is fun. I want you to hold up whatever you're reading or writing on and put it in front of you. And I want you to nod your head like no. Don't go too fast. Can you still read the stuff on your paper? Yeah, okay. Now, wait, we're not done. Now I want you to take the paper or computer or whatever, and I want you to move it. Can you read it? Not very well, right? It's a lot harder. Okay, so why is that? You can actually do the same frequency and amplitude of movement of your hand versus your head, and in one case you can stabilize your eyes, in the other case you can't. The reason for this, and you have access to the motor commands for both. The reason is that you actually need this really fast reflex, which is called the vestibulo-ocular reflex, and what it does is it counter-rotates your eyes exactly the same amount that your head moves. So this is like super critical. If you're walking along and you want to read a sign, your eyes are being held steady even though your head's bobbing all around because of this reflex. So you can imagine that if you lose the calibration on this reflex, if the gain is wrong, so your eyes don't move far enough for a head movement or they move too much, that everything's going to look like it's jumping around. Do any of you remember when video cameras didn't have the auto stabilization thing? I do. And videos, home movies looked like crap, right? Because everything bounced around. That's what it would look like if you didn't have a vestibulo-ocular reflex. So, ta-da, the cerebellum keeps it calibrated. And if you damage the cerebellum, you lose that calibration. Okay. Okay, so this is just a cartoon drawing just to show you that when you damage the midline cerebellum and specifically the vestigial nuclei, if you do this in a monkey, they'll fall, and they'll fall to the side of the lesion when they walk. but they will be able to do very fine motor things and reaching just fine. So that control is really only for that kind of movement. Likewise, if you damage the midline, they'll have trouble with reaching, grasping. And when you damage the lateral cerebellum, the dentate, the only motor problems you get are really very fine motor problems, and then probably cognitive problems that are a lot harder to test in monkeys. So you do get very specific problems. Okay, so what time is this class over? 2:30, right? 2:20. Okay, sorry. Don't worry, I won't go over. Okay, so what I want to talk about now is I want to go into a little more depth in the cerebellar cortical circuit. And I'm going to simplify it greatly, but first I want to tell you about the cell types and layers that are in the cortex. This is a chunk of cerebellar cortex that's been pulled out. This is out of Candel. And what you can see in this little block of cerebellar cortex are the cell types that are found in the cerebellum. In the granular layer, which is the deepest layer, are little tiny granule cells. And these little tiny granule cells are packed in there. They're these guys. They receive all of the inputs coming into the cerebellum. So granule cells get all the inputs from all the different places that we discussed. It just depends on where you are in the cerebellum, what kind of information it's getting. But the granule cells then send up an axon that branches like a T, and this branches into something called a parallel fiber. So this is receiving input, and this is the cell type that's so dense in the cerebellum. This is what makes the cerebellum have so many cells, is there are tons and tons and tons of granule cells, and it's receiving input into the cerebellum. Now this parallel fiber branches out, and it actually excites this other cell type that sits in the Purkinje cell layer, which is named a Purkinje cell, making it easy for everyone. So this Purkinje cell is a really cool neuron. It's got this huge dendritic arborization. It looks like a fan. And actually, if you look at it from the side, it really does kind of look like a C fan. So you look at it from the side, and it's very narrow. They've tried to diagram it here that way. And it receives lots and lots and lots of inputs from parallel fibers. So the Purkinje cell is receiving parallel fiber input, and it's the output of the cerebellar cortex. It projects down to the cerebellar nuclei. So the input comes in here, and then the output goes out through the Purkinje cell. So that's like most of the circuit that we're going to talk about, sadly. The rest of the neurons, the other 3 types of neurons in the cerebellum, are all inhibitory interneurons. There's a stellate cell, a basket cell, and a Golgi cell. And these 2 sit in the molecular layer up here, which is the 3rd layer, and this one sits down here in the granular layer. We don't really understand what those inhibitory interneurons do. We have some ideas. But for this class, I'm going to ask you just to know something about Purkinje cell processing. The other thing I didn't tell you about the Purkinje cell is that it gets a single input from a part of the brain called the inferior olive. And this single input climbs up the Purkinje cell, and it's called a climbing fiber. So it gets lots and lots and lots of parallel fiber inputs, and each Purkinje cell only gets 1 climbing fiber input. And so you have this circuit where you have all this information coming into the cerebellum, and it's going to these granule cells, which branch into parallel fibers, impact the Purkinje cells, and then you have this 1 climbing fiber input that comes in, and when it activates the Purkinje cell, it really activates the Purkinje cells. It causes a very complicated action potential waveform that has lots of little wavelets. So I'm going to tell you more about that in, actually, you know, I usually tell you about that. I usually go right into my cerebellar cortical studies, but I think I've obviously rearranged this, so I think I'm gonna come back to this in just a minute, but keep in mind cerebellar cortex, and we'll talk about its function in just a minute. Okay, now if we think back though, and think about the output of the cerebellum itself, so the deep cerebellar nuclei, you know, maybe I'll go forward and do this. Okay, I'm going to keep going on the cortex. So if you think about the cerebellar cortex, what I've done basically is I have pared it down to this circuit. You've got a Purkinje cell sitting up here, and you've got mossy fibers coming in, and they're projecting to a granule cell, which gives rise to a parallel fiber. And then you've got a climbing fiber coming in and projecting to the Purkinje cell, and it only gets one climbing fiber input. It gets lots of parallel fiber inputs. This is a deep cerebellar nucleus. It could be any one of them. And it gets input from both of these things that are excitatory, and the Purkinje cell's input to the deep cerebellar nucleus is inhibitory. So this is actually a pretty simple circuit. You get two kinds of inputs. Both go to the nuclei. One is a climbing fiber. One is a mossy fiber that impacts this granule cell. Purkinje cell then inhibits the nuclei. And the important thing here is the balance between the inhibition and the excitation in the deep cerebellar nuclei. That's what sets the firing rate. And so somehow changing the circuit allows you to change your behavior and causes these problems that I showed you earlier with these patients. So causes the ataxia. So one thing that we think is very important in the cerebellum is a specific type of learning process. Now, I'm going to show you a little example of this, and I'm going to show you specifically what happens in the circuit, but to understand it, we have to think more a little bit about Purkinje cells. When you record from a Purkinje cell, so if you extracellularly record from a Purkinje cell, what you get is a train of action potentials, and that's what's shown here. On the top of this is a neural recording, and you have lots and lots of action potentials from a single Purkinje cell. They fire at about 100 hertz. Every so often, though, in this trace, you see sort of these positive-looking blips. So these are all the regular action potentials, and then every so often you see something that looks like this. So these action potentials we call simple spikes, and they are driven by the parallel fibers. These action potentials here that occur much, much less often are complex spikes. These are driven by the climbing fibers. So if you have both, this is what they look like together. Now, if you're trying to control a movement, ongoing movement, 100 hertz is not bad at all, because your movements are typically pretty slow. They're usually between 2 and 5 hertz, 1 and 5 hertz, below 5 hertz. Complex spikes fire 1 time per second, 1 hertz, maybe 4 hertz, not very often at all. So that kind of information wouldn't be very useful for guiding online movement control. And so what people have hypothesized and actually shown a lot of compelling data for is that the complex spike is really a teaching or error signal, and it changes how the Purkinje cell responds to parallel fiber inputs. So it changes how excitable that Purkinje cell is to simple spike, to parallel fiber simple spike types of input. And so this learning actually turns out to be very important for all different kinds of movements. And I'll show you the simplest form right now. And then we study very complicated forms of learning that are cerebellum dependent, that involve the whole body and walking and integrating vision. But this is the simplest. How many of you have heard of classical conditioning? Everybody. Good. So this is a very common paradigm. It's called eye blink conditioning. And so this is usually done with rabbits. You can do it with other animals, but rabbits have been sort of the most commonly used paradigm. If you sound a tone, the tone is meaningless to the rabbit. It may startle it a little bit, but it's pretty meaningless, because it's not associated with anything. But if you pair a tone and then immediately give a shock to the side of the eye, just a little shock, the rabbit will blink. And eventually, if you sound the tone, the rabbit will blink in anticipation or right as the shock is occurring. So it learns an association between the tone and the shock. And so it causes this anticipatory eye blink. Now, we know that this depends on the cerebellum, as it turns out. What we think is happening, and this is based on lots of studies, and this cartoon is showing sort of a diagram of it, is when the tone comes in, its projection via the pons activates mossy fibers that go and activate granule cells. And those granule cells turn on the Purkinje cell. Now, the Purkinje cell inhibits the nucleus. This projection excites the nucleus. And you can imagine if everything were balanced, it would just sort of cancel out. And the tone wouldn't do anything. But as you learn, as you hear the tone and start to get this shock, which the animal we think perceives as an error or something unexpected, it causes a climbing fiber to be activated at the same time these parallel fibers are activated. When that happens, it leads to a kind of learning called long-term depression. And so the synapses between the parallel fibers and the Purkinje cell that are active at the time that the climbing fiber fired get depressed. They lose efficacy, so they don't cause the Purkinje cell to become as excited. So if you do this over and over and over again, this input now drives this Purkinje cell less. And so the Purkinje cell inhibits the deep nuclei less. If that's the case, then the tone now, through its direct projection, can cause a blink to happen. And it actually does this through some brainstem nuclei. So what's happening, just to think this through again, is that you're getting a discrete error signal, and then on the next trial, we have depressed the parallel fiber Purkinje cell synapses. And that, in this case, leads to the development of this conditioned response. Now, this is a very pared down, simplified form of learning, but it's been very useful for studying these kinds of processes. Since this work it's still going on, has been done, other kinds of work has been done to relate this to much more complex movements. Eye movements, pursuit eye movements, reaching movements. And so we think that this system of error feedback on one trial, changing how the cell responds on the next trial, is important for motor learning. Okay. Now, I'm going to go back and talk at the end of this lecture about the output of the cerebellum. So we were just talking about how the cerebellar cortex is plastic. It gets changed with error signals, with error information, and then eventually you get a change in the output of the cerebellum. The cerebellar output from the deep nuclei is excitatory, and as I mentioned before, it projects out to other motor structures to help control movement. There is a somatotopic map in the nuclei, and as I told you before, different parts project to different parts of the brain and to the periphery. Importantly, deep cerebellar nuclei activity is before movement. So the motor cortex comes on basically first, then the cerebellar nuclei, and then, or simultaneous with that. So we think it's some kind of internal loop. And eventually muscles are activated. So muscles are activated the latest, and a movement is initiated. So at the beginning of the class, we talked about why you need an internal feedback loop, why you need an internal model of movement, because of time-delayed feedback and because of a complicated plant that you have to control. And so the question that people have been asking more recently is, are there any patterns of neural activity in the cerebellum that are consistent with this kind of model? And so you'll remember that we had a forward model and an inverse model. And so there has been a study recently done by Tim Ebner's group asking the question of whether you can see a signal that looks like a forward model. So a forward model, just to remind ourselves, is a model that would take a motor command in and it would send out something about the predicted kinematics, the predicted action that would be taking place. And so what he did, I think, was pretty nice. He had a monkey learn to make a movement where he had to track something like that was rotating like this. And what he did, and we don't need to worry about this part of the panel, but here are some traces of the monkey tracking. Now, the cool thing that he did is he had the monkey do this tracking movement, sometimes with a viscous force, sometimes with no force, sometimes with an elastic force. So the monkey had to make all different patterns of muscle activity to make the same kinematic movement. And you can see here that the kinematics of these movements under all these different loading conditions are different, are the same. The load is different, the muscle activity is different, but the kinematics are the same. So if you think about this, what he's looking for is a lot of different motor commands are leading to the same kinematics. So he's looking to see if you see anything that looks like a forward model output. Does the activity relate to the kinematics, or does it relate to the forces? And what he found is this is neural activity across a couple of different neurons. And what he's showing is the position of the monkey's hand. And this is the rate of firing of the neuron. These are Purkinje cells. And these are all different loads. So what you should notice here is that the cell fires the same way no matter what load. It's always the same. And so what that suggests is that that output is more like a forward model, where it's telling you something about the expected kinematics. Because in all different load conditions, the monkey is still going to make the same kinematics. And so he suggested that that was evidence of a forward model in the cerebellum. Now, of course, everybody who does these experiments often tries to find what they're looking for. And if you go back, so the interesting thing about this study, by the way, is this clear to everybody? Does this make sense? Or is it like, do you want to go through it again?
Yeah, can you just say it one more time?
Yeah, absolutely. Okay, so I'm going to say it without the slide. So if you have a forward model, what happens is that motor cortex is going to send some commands to the arm. And internally, those motor commands are going to be evaluated. And it's going to send back a prediction about where the hand's going to go given those motor commands. Now, if I'm in water or if I'm in air, for example, I might have to make different motor commands, but I can predict, given the environment, that I'll still go in the same direction. So that's the kind of activity they saw in the cerebellum. They didn't see activity that changed with the motor commands. They saw activity that looked like what the animal was going to do. And so that was a suggestion that this looked like a forward model. Did that help?
Yeah.
Okay, good. So but that was in the cerebellar cortex. That was in the Purkinje cells. And you could argue that Purkinje cells are getting all this afferent input from the periphery, so that could just be an afferent signal. That could just be feedback. So a long time ago, Tom Thatch did a study that is actually really cool. What he did is he taught an animal to make a series of movements where he moved his wrist, and I'll move my arm since it's bigger, and he had to move to 3 positions in this direction and then 3 positions in this direction. And what he did is he loaded the animal. So when he was moving in this direction and starting to go back, he always had to use these muscles. And then he could load the other side, and he'd have to use these muscles. So what he could do is he could make the animal go in both directions using the same muscle. Or he could switch it up. And so that's kind of what's diagrammed here. So for position 1, 2, 3, and 4, the muscle that this animal had to use was always extension muscles. And then he repeated it using flexion muscles. Now, if the neurons care about the muscles, which would be like an inverse model, then you should see neural activity that looks like this. And if the neurons care about the kinematics of the movement, which was the output of a forward model, then it should look like this. Okay? So again, forward model output is kinematics. Inverse model output is force or muscle activity. And so he could discriminate between those two things, even though back then that's not what he was looking for. And what he found in the deep cerebellar nuclei is if it looked like a muscle, it would look like this, and it would be highly correlated to the muscle activity. And if it looked like joint position, the kinematics, it would look like that. Each of these dots is a neuron. And so if a neuron's sitting down here, not related to joint position, but very related to muscle, this is an inverse dynamics looking neuron. And if a neuron's sitting up here, it's a forward dynamics looking neuron. Now the bottom line of this, sadly, is that you have a lot of both. I do not think that there is one particular flavor of model sitting in the cerebellum. My hunch is that you have both of these things. And you can imagine that you might need both of these things because this cerebellum gets inputs about intended movement direction, and it might want to help you predict the right motor commands, but it also gets information about intended motor commands, and it might want to be able to predict where you're going to go. And so when you read papers, this is what's so interesting. The Ebner paper was very, very clear. It was like published in Nature Neuroscience, and it said, this is what this is. And this is a paper from 1978. I mean, that was probably quite a bit before any of you were born. Maybe your parents were born. But I mean, this is what he was doing in 78. That's amazing. And what he's showing is actually very, very relevant to this. And this was I think this was published in Journal of Physiology. So I think that what's so clear about these things is that, first of all, we think that these models do exist. And we think we see some neural correlates of it. But at this point, we don't think that, or at least I don't believe that there is consensus about what kind of model is being encoded in this neural activity. But nobody's looked that well. So these are like two of the studies that have been done that are convincing. OK, since I think the last thing I'll tell you in the remaining 3 minutes is that neural activity in the cerebellum is modulated much more for complex movements than for really simple movements. So in this case, if you look at the green circle, these are histograms from neural recordings where they taught a monkey to do lots of really simple movements, like a single finger or single wrist or single elbow. And then they had the monkey do this whole arm thing, which is the reaching circled in red. And many neurons in the cerebellum don't modulate very much for the simplest of movements. But as soon as it starts to get complicated, they care. And they start being active in relation to that movement. So you remember what I told you early in the lecture. You need a cerebellum to make predictions about the complex mechanics of your body. And so if your cerebellum is having to learn how to activate muscles in your leg in anticipation of forces generated by your arms, then this kind of makes sense, that the neural activity may not care as much when something is really simple and not having mechanical consequences or these coordinative consequences. And so what's cool, if you look at patients, the patient I showed you who was doing the finger to chin movement, so whenever you examine a patient, this is what they try to do. They try to do this. If you put your finger out, they try to do it like this. Now you can see what I'm doing, right? I've taken my arm and I've locked it up against my body so I can make a very simple movement. And I mean, I can see people in the airport who I know have cerebellar ataxia and they're not drunk because when they're doing something, they do it like this. You know, they figure out ways to stabilize themselves when they brush their teeth or when they're eating. And that's because they have lost this multi-joint control, which we think is reliant on these kinds of predictions. OK, I think I'm going to stop there. And next time, we'll talk about the basal ganglia.